So hello everyone, my name is Danilo Amaral, and today I'll bring to you our recent findings about the transdermal data in clade with rapid speciations, the clade Cereus fernambucensis, which is a cactaceae species that are present in the Brazilian Atlantic forest. And I really would like to thank you, the organizing committee, for the opportunity to come here today and discuss a little bit with you. So probably you already saw those pictures. They represent the species richness in the world, and most of these species diversity is located or near to the neotropical region, which display a very interesting conditions associated with several abiotic changes and diversification. And those conditions is still a challenge for us that work with evolutionary biology. So based on that, we checked and bring to you a very interesting neotropical region, Brazilian Atlantic forest, which is a bit a large range of abiotic conditions. As you can see in this picture right here, they display several distinct physiognomy around the distribution that comes from the south of Brazil to the northwest of Brazil. Besides that, this region suffer with distinct different conditions during the Pleistocene climatic oscillation. The species that you have several events of physical barriers, such as some rivers and mountains, and also this area displayed several neotonic events in the last million years that we suggest to be a major cause of the diversification in several species in this area. And for us, as I told before, evolutionary biologists, we are interested to understand a little bit about these events that promote diversification, but we also try to understand some correlationship between genotype and phenotype, mainly associated with rapid speciation events. And for that, today, I bring to you the clade Cereus fernambucensis, which is a cactaceae species widely distributed in the Atlantic, in Brazilian Atlantic forest from the southern portion to the northwestern portion. These clades comprise three main species. One is Cereus cerucifer, which is occur within the southern portion of Brazilian Atlantic forest. You have the Cereus fernambucensis species, which occurs from the southern of São Paulo to the northwestern of Brazilian Atlantic forest, and the species Cereus insularis, which occurs in Fernando Noronha Island. Yeah? And these three species, besides, as you can see here, that display distinct geographic separation, they also display morphological difference and also climatic niche changes, which should be interesting to study the genome among the populations to try to observe some possible signature of evolution in their genomes. Based on that, we try to observe if abiotic and biotic changes may promote speciation. As we already know, speciation derived by a combination of factors. So if you have new ecological opportunities, such as changes in elevation or changes in climate, such as precipitation or temperature, and with this species display some genetic variation that can be response to the abiotic change and promote some traits in a lineage specific, we can also increase the speciation rate in this group. And nowadays, with the advancing of omic approach, as I said before, the transcriptoma data, we may exploring new molecular data to gain insights about the mechanisms that promote this species diversification, may associated with neotropical region. So here in these studies, you seek to understand the evolutionary forces that drive their morphology, distribution, and genome. Try to address some questions include the contribution of natural selection at interspecific level associated with possible evidence of positive selections. So the species of Cereus clade has a colonization distribution from south to north, become a very interesting wide distribution group from Einzelberg areas around 1 million years that has a geographic extension from Restinga, and after that reached the Fernando Noronha Island around 80,000 years ago. For our studies, we choose 7 populations from Cereus species of these clades, 2 of populations of Cereus certifer, 4 populations of Cereus fernambucensis, and 1 population of Cereus insularis. We performed transcriptome sequence assembly, which observed around 93,000 transcripts per population and around 94% of completeness, which means that you have a very good assembly quality. After that, we perform some transcript abundance annotation, identify some orthologous protein, which will observe around 1,000 single copies genes. And with using these genes, we perform the NDS rates and a positive selection genes. After that, we also use a phylogenesis approach, which means a genome-wide association, using a phylogeny as a background for that. When you annotate these genes and perform some transcriptional analysis, we observe that most of the species and population share common GO terms. That means that they display similar metabolic pathway. In this case, probably all these common genes have the same physiological and biochemical homeostasis important for those group of species and also populations, and are promoting the viability for these organisms to this the current niche condition. However, we still find some signature of positive selection genes and high DNDS rates in some specific lineages or clades. For example, here, when you observe the movement from south to north, we have the first high DNAS change in the clade 4, which is the first incursion of Cereus fernambucensis from south to north region. And those genes associated with positive selection in these clades are associated with light signaling and also response to light stimulus. We also have some changes in the clade 7, which represents the peripatric speciation of Cereus insularis, and we have genes in those clades with PST related to genes of thermal, oxidative, and drug stress, and also photosynthesis metabolism. Based on the results, we believe that most of them are physiological and molecular processes that can be involved with the resilience under abiotic stress in those groups. When we perform our phylogenesis approach, we observe some abiotic and biotic pressures in those species, and they are associated with mainly with 2 major metabolic pathways: biosynthesis of secondary metabolites and energetic metabolisms. And when we are looking for some traits, abiotic and biotic traits separately, we observe that we have some longevity regulation pathway present in reproductive traits, pressure, and also when you choose a precipitation abiotic trait, we observe some plant and pathogen interaction and cellular process information. Based on that, the geographic transition and expansion in this group, as well as the initial shift that can be occur when you have the change of domain among those species, mainly associated with the transition from an Inselberg area to a Restinga, and after that to the Restinga to an island condition, you probably observe some correlation between genomic and those traits. As I told before, abiotic traits are observed including synthesis of secondary metabolites, interaction with plants pathogen, and energy genetic metabolism. And when you check the biotic traits, you observe some genetic information process and aging. So based off that, the physiological and molecular process associated with those traits can able to allow this species to adapt to those new conditions in the geographic and niche shift. So, pessoal, this is all. I would like to thank you, everyone, for the opportunity. Thank you, the organizing committee, all collaborators and author of this work, also financial and logistic support for the opportunity, such as FAPESP and Universidade Federal do ABC. And I see you soon. Bye-bye.